University of Liverpool, and has been doing a lot of work in the long-acting injectable space, and particularly HIV and emerging economies. He's a chair of the British Society of Medicine, among a number of other activities. Good morning, Renee, or good afternoon. He does clinical and basic research on mechanisms in pharmacokinetics and pharmacodynamics, as well as looking at his own technologies and long-acting injectables. And Andrew is a great introduction to this whole field.
So I'd like to extend a big thanks to the organizers for organizing this workshop today, and for inviting me to present on established and new technologies for long-acting drug delivery. So I'll start by saying a little bit about what I consider to be the arc of benefits for long-acting drug delivery. And this is adherence, retention, and completion. Back in 400 BC, Hippocrates was quoted as saying, "Keep a watch also on the faults of patients, which often make them lie about the taking of things prescribed." So I think it's fair to say that adherence to medication has been a problem for quite some time. From a pharmacokinetic perspective, of course, the goal of therapy is to administer the drug to patients. When this is done via the oral route, plasma concentrations rise to Cmax before subsequently being cleared and administered again until the patient reaches steady state pharmacokinetics, and the goal of therapy is to maintain the plasma concentrations within this therapeutic range. But of course, patients aren't always adherent to medication, and this can have profound consequences for the pharmacokinetics and ultimately the efficacy of the medicine. In some cases, a patient might miss several doses of the medicine, in which case they need to be dosed right back up to steady state. And in other cases, patients just cease taking their medication altogether, or may occasionally take a dose of their medicine. So the goal of long-acting drug delivery is to provide a single administration of the drug which maintains the plasma concentrations within this therapeutic range over a protracted period of time. So oral delivery, I think it's fair to say, places success of therapy in the hands of the patient, whereas long-acting drug delivery has the potential to be transformative for patient management, not least because of the benefits for adherence to medication, which can be partially or entirely mitigated. But it also has profound benefits for retention of patients within therapy programs and for indications within the range of duration of exposure which is achievable. Completion of a course can be completed from a single clinic visit, and this can have profound benefits in low and middle income countries as well as in high income countries. So what are the long-acting technologies? Well, there's several of these. Probably the most successful approaches have been injectable drugs or implant type approaches. And we've got emerging technologies such as microarray patches, which are being quite intensively researched for long-acting drug delivery, but also other approaches, particularly within infectious diseases, such as the broadly neutralizing monoclonal antibodies, and then approaches which are emerging such as gastric resonance devices, which hold the drug within the stomach and gradually release drug for oral absorption, or other technologies for things like HIV, such as the vaginal ring type technologies, which provide protracted exposure to the drug locally over a long period of time. So I'm going to focus mainly on the technologies on the right hand side of this slide for the sake of this presentation. So before we start looking at the specific technologies, I think it's important to acknowledge that drug selection is critical, as is technology selection, and this must be driven by what you are hoping to achieve from the delivery modality. Potency or target plasma concentrations and dose ultimately set the bar and define the duration of achievable exposure. It's clear that drug selection is key. Long-acting delivery is only achievable for highly potent drugs, which therefore have very low target exposures, and really this sets the bar above which you want to maintain those concentrations. Dose is ultimately capped by the tolerability and the practicality, and is heavily influenced by the technology and the desired route of administration. And I think probably most importantly, drug loading within your final format is a key component of success for any long-acting approach. Some technologies require a high mass of excipient or carrier material to control drug release, and the mass penalty must also be acknowledged when using prodrug derivatization in order to improve compatibility of a drug with a particular technology or device. So long-acting injectables have been around for quite some time. The first long-acting injectable was actually for estrogen, reported in the literature in 1953. Since then, there's been a small stream of publications on long-acting injectables, which was only really at the beginning of the 21st century started to gain momentum with development of things like medroxyprogesterone acetate for contraceptives, and some of the antipsychotic agents such as paliperidone palmitate, and most recently the emergence of new treatment modalities and prevention modalities for HIV with the advent of cabotegravir LA and rilpivirine LA. So the first of the technologies that I'm going to speak about are the aqueous suspensions. There's examples of this type of technology for intramuscular and subcutaneous administration, and I think it's fair to say that the aqueous suspensions to date have been the most successful injectable approach. There's numerous examples for schizophrenia and contraception, as well as the newer antiretrovirals rilpivirine and cabotegravir. And this technology achieves the highest drug loading of all the existing technologies, and is better tolerated than some of the other technologies such as the oil depot formulations. The medicine itself constitutes a suspension of drug or a prodrug as particles, which can either be measurable within the nanoparticle range or in the low micron particle range. Excipients require very careful selection, but drug release is controlled usually by drug solubility and the biology of the depot site in order to slow down and protract the drug release into the systemic circulation. There's lots of technologies available for producing these nano or microparticle suspensions. These include things like the emulsion-based methods, nanoprecipitation, high pressure homogenization, but the most successful to date has been nanomilling, where you literally take big chunks of drug and smash them up until they're in the nanoparticle size range. Another technology which has been used quite extensively is the oil depot formulations. The existing oil depot formulations involve intramuscular delivery and provide exposure between 4 and 12 weeks after administration, depending on the specific drug and indication. High drug loading is usually achieved through formation of a prodrug to increase the solubility within the oil. So typically you take a drug and add a very large lipophilic prodrug moiety in order to really increase that solubility within the oil. And commonly used oils include sesame, peanut, castor or coconut oil. And excipient selection is usually based on enhancing solubility, reducing viscosity, or in some cases providing local anesthesia. Oil depot formulations have proven successful. They are clinically used, but they must be administered slowly usually, and they're very commonly associated with injection site pain that in some cases can persist up to 3 months after administration. Another technology which has been successful are the polymer microsphere technologies, usually administered intramuscularly as a microparticle suspension. They're compatible. One of the interesting things about this technology is that they're compatible with water-soluble and water-insoluble drugs. Usually though, the drug loading is lower than for some of the other technologies because a lot of the final format is composed of the polymers which are required to control the drug release from the formulation. Usually soon after the polymer microsphere is administered, you get an initial release of drug at the surface of the polymer, with subsequent hydration needed. It may mean very low drug release over the first weeks after administration, and then after that hydration of the formulation locally, the release then occurs through gradual erosion of the polymer, followed finally by bulk erosion when most of the drug has been released from the formulation. Another technology which is emerging, exciting technology, are the in situ forming gels. Typically with the in situ forming gels, you take an organic solution of polymer which is combined with a solution or a suspension of drug particles. It's again another technology which is directly compatible with water-soluble or insoluble drugs. And that organic solution of polymer, a drug solution or suspension, is injected into the depot site, and depot formation is then triggered by polymer precipitation, usually by some physiological trigger such as aqueous insolubility of the polymer. You get cross-linking of the polymer, which then subsequently traps the drug or the drug particles within the depot site, and subsequent erosion of the polymer again controls release from the depot site into the local and then ultimately the systemic compartment. So those are the injectable technologies in a broad sense. Implants and devices are clearly exciting technologies for long-acting drug delivery as well. Here's just some systemic delivery examples in contraception. I think it's fair to say though that the implants and the devices require really highly potent drug because most of the format which is administered is composed of the implant or the device itself. However, of all the technologies, the implants are really only the only reversible approach to systemic long-acting delivery, and that's important because if a patient experiences an adverse drug reaction or if there's a lack of tolerability, then it's important to be able to remove the approach. Biodegradable implants also exist. And there's lots of examples of implants for localized drug delivery, for example, intratumor, intraocular drug delivery, and there's much recent interest in the development of antiretroviral implants for prevention as well as treatment strategies, although these are mainly focused on highly potent NRTIs such as tenofovir alafenamide and EFDA. Just wanted to acknowledge that there's a lot of interest in other drug delivery type devices such as gastric residence device, which is administered as a capsule which subsequently springs open within the gut to reveal these branches on the gastric residence device, which are capable of gradually releasing drug into the stomach, which can then be absorbed through the intestine. Another type of implant which I've seen in the literature are these so-called osmotic engine devices, where you have a compartment filled with drug coupled to what's termed an osmotic engine. And this osmotic engine draws in fluids from the surrounding tissues, which subsequently push a piston which ejects the drug gradually out into the local environment. And there's some interest as well in more electronic based solutions such as wirely control devices, one of which is a microchip, termed the microchip implant, where you have these compartments of drug within the implant, which can be with a polymer matrix, which can be eroded through an external signal from the body. Another emerging approach for long-acting drug delivery, which is being discussed quite frequently now within the literature and at meetings, are the microarray patches, or the MAPs. These are patches which are administered by placing on the skin of the patient. The microneedles themselves are composed of a polymer such as polyvinyl alcohol, which is mixed with a long-acting injectable formulation. So, for example, rilpivirine long-acting formulation and nanoparticle formulation mixed with this polymer to create the microneedles themselves. And this is cast upon a base plate which consists of another polymer such as polyvinyl pyrrolidine. And the MAP needn't, I think it's important to recognize the patch itself needn't be worn for long term for drug delivery, because when the patch is typically worn for an hour or so, and then when the patch is removed, the microneedles remain in situ and gradually erode to release the drug to provide the long-acting drug delivery. The approach has been postulated as a means of delivering long-acting formulations via a different route. And I think this is an important subtlety, because what's being explored at the moment is taking a classical injectable formulation and using the microneedles to deliver that formulation. So in many ways, this isn't necessarily a long-acting formulation or technology approach. The microarray patch itself really replaces the syringe in order to and the needle in order to provide the long-acting drug delivery. The approach is clearly expected to exhibit better tolerability and mitigate the need for trained healthcare workers, which can be important in many contexts. And it aims to deposit the formulation as a depot in the skin with a subsequent dissolution into the interstitial fluid. At least in one example, 7 days of therapeutic exposure was demonstrated preclinically in animal models. So I do want to say a little bit about the broadly neutralizing monoclonal antibodies. And in this case, you're just going to specifically use the HIV example. These are different. It's different from the other technologies that I've discussed, in that this is more of a biological engineering technology, and it really generates passive immunity in the case of HIV by disruption of the interaction between the envelope spike and the CD4 receptor. For HIV, there's at least 9 broadly neutralizing monoclonal antibodies which have been developed or are in clinical development. They all target various regions of the CD4 binding site, the V3 loop, the V1, V2 loop, or the GB41 membrane proximal external region. Now, this approach clearly has potential for therapy, but also in prevention of HIV and other infectious diseases. It has been shown that when single BNAbs are used, resistance can emerge relatively quickly, and so most of the ongoing programs now are looking at combinations of BNAbs which targeting non-overlapping sites within their target. Now, what makes this a long-acting approach is that changing specific BNAb amino acids increases binding to the neonatal Fc receptor, or FcRn, which promotes endosomal recycling of the antibody and therefore dramatically extends the serum half-life. So accordingly, the BNAbs have been developed for once every 2-month administration, and are looking really attractive. So just to say a little bit about this endosomal recycling, which extends the half-life of the BNAbs. Well, the antibodies are taken up into the cell via these endosomes, and within the endosome they're able to they're able to bind to the FcRn receptor. And the consequence of this is that the antibodies bound to the FcRn are targeted for endosomal recycling and re-enter the circulation, whereas those which are not bound to the FcRn are degraded by the proteasomal degradation which occurs within these intracellular organelles. So specific amino acid changes within the Fc region increase the FcRn binding affinity of the antibody, and this is the technology which is applied in order to extend the half-life and what makes these long-acting approaches. And this isn't real data, this is just representative data, but really profound increases in the half-life of the antibody have been demonstrated by changing these specific amino acids within the antibody. So no presentation, I think, at the moment would be complete without some mention of the COVID-19 pandemic. And specifically, I really just want to raise the question of whether we can bring forward rapid long-acting interventions for COVID-19. Long-acting medicines, I think it's fair to say, would buy much needed time for vaccine development for SARS-CoV-2. There's a huge potential opportunity for pre-exposure prophylaxis to protect healthcare and other key workers within the supply chains. And there's been already quite significant recent interest in BNAbs for SARS-CoV-2, which are at various stages of development, and again combinations of BNAbs which target overlapping sites within the coronavirus. For small molecules, we really need long half-life drugs with these highly potent anti-SARS-CoV-2 activity. We have recently published a paper which outlines the known potency of a number of repurposing drugs which have been investigated so far, and we've got a database in Liverpool where we're keeping this up to date. So if anybody does have interest in this area, then please feel free to drop me an email. So just to summarize long-acting technologies, there's no real classification of long-acting technologies yet, but if we start to draw one out, I think it's fair to say you can split long-acting therapies into devices or into the parenterals, the injectables. Within the devices, there's emerging technologies such as the vaginal rings, the gastric resonance devices, microarray patches, as well as the established implant type technologies. And in the injectables, we have the oil depot injectables, the aqueous particle dispersions, we have the in situ forming gels, the polymer microspheres, and the monoclonal antibodies. Just a caveat is that some literature defines polymer microspheres and in situ forming gels as as implant type technology. So I think we have got some work to do in sorting out the nomenclature as this field develops. So just to finish off, there's multiple technologies being explored for long-acting medicine delivery across different indications. Ultimate technology selection is going to depend upon drug physical properties as well as the pharmacological properties. As opportunities for reversibility in drug delivery are only currently afforded by certain implant technologies, but longer exposures require higher potency than available for many drugs. With these technologies, and the aqueous suspensions have proven the most successful technology to date. They have the highest drug loading of all the technologies. They're better tolerated than some of the technologies such as the oil depot formulations. They are restricted to water-insoluble drugs, but we and others are exploring prodrug derivatization to overcome that for some small molecules. And this type of technology may also be compatible with delivery via a microarray patch. So I'll just leave you with some acknowledgements to the full team in Liverpool, various funding agencies, and some of our superb international partners. Thank you.